Welcome to the deep dive. Today, we're embarking on a journey, really getting into one of the body's most efficient, intricate, and often unsung heroes, the small intestine. It's truly the critical hub for how we turn a meal into fuel and, well, the very building blocks of life itself.
It is. It's a finely tuned system, truly. We're
Yeah.
talking about, precise control over everything from breaking down these really complex molecules to carefully managing how much fluid moves in and out. For anyone looking to master human physiology at, let's say, an elite level, understanding the system is absolutely fundamental.
And that's exactly our mission today. We've got a detailed physiology chapter as our source, laying out the entire small intestinal phase of your body's response to a meal. We're going to extract every essential piece of knowledge, uncover the surprising facts, and ensure you walk away with a truly comprehensive understanding, the kind that, well, elite professionals possess. So, to start us off, what is the small intestine all about? What's its core identity?
Well, at its core, the small intestine is where nutrient assimilation primarily happens. This is the place where food, now this semi-liquid mixture called chyme after leaving the stomach, blends with various secretions for crucial digestion and absorption. Its design is just a marvel of efficiency. It's remarkably long, right? A coiled tube, but its secret weapon is really its internal architecture. Think of it as having multiple layers of specialized folds, like the large folds of the mucosa and submucosa, and then even smaller sort of finger-like projections called villi. And on top of those villi, each epithelial cell has these microscopic brush-like projections called microvilli. All these things together dramatically increase the internal surface area, immense capacity for digestion and absorption.
That massive surface area, wow. That's just the beginning of its genius, isn't it? And one of the defining features of this entire small intestinal phase you mentioned is this incredibly controlled delivery of chyme from the stomach. It's all about perfectly matching nutrient delivery to the intestine's, well, immense digestive and absorptive capacity. So, okay, let's trace the meal's journey. How does the stomach, which can comfortably hold, up to a liter of material, how does it decide when and how much to send down to the small intestine? It can't just dump everything at once, right?
And that's a critical question. How does the system prevent overwhelming the small intestine? Good point. The rate at which the stomach empties its contents, it's highly dependent on the meal composition, specifically its macronutrient content and whether it's liquid or solid. Liquids, for instance, empty quite rapidly. Solids, however, they have a distinct lag phase because they must first be broken down into particles smaller than, say, 2 millimeters before they can even pass through the pylorus. That's the muscular valve at the bottom of the stomach.
Right, the pylorus. So there's this intricate dance of coordination. What does that look like when a meal first arrives in the stomach?
Well, initially after a meal, the stomach is largely relaxed. It's mainly engaged in mixing its contents, and the pyloric sphincter is mostly closed. So not much emptying occurs right then. But then to actually initiate emptying the upper part of the stomach, the proximal stomach, it increases its muscular tone. Simultaneously, contractions in the lower part, the antrum, they strengthen. This is what we call the antral pump. Then the pylorus opens, and then critically, contractions in the duodenum, that first part of the small intestine, are inhibited. This allows the semi-liquid chyme to just flow down a pressure gradient right into the small intestine.
Okay, that makes sense. Flowing downhill, essentially. But once the chyme hits the small intestine, surely there's a feedback loop to prevent too much from entering at once.
Absolutely. As chyme enters the small intestine, a really sophisticated feedback system kicks in. Specialized sensory neurons, predominantly vagal ones located in the intestinal wall, they detect the presence of nutrients, acid, and highly concentrated or hyperosmotic content. This triggers a reflex, sends signals back up to the stomach via vagal efferent pathways. And the result, gastric emptying slows right down. This happens by decreasing those antral contractions, causing the pylorus to contract and close more tightly, and by reducing motility in the proximal stomach. That lowers the pressure inside the stomach. And this same pathway, interestingly, also helps inhibit gastric acid secretion when the duodenum is already busy with nutrients.
So it's truly like a sophisticated traffic controller constantly adjusting the flow rate. And the hormone cholecystokinin, CCK, that plays a pretty substantial role here, doesn't it?
Oh, it's vital, absolutely. CCK is released from specialized endocrine cells, called I cells, found right there in the duodenal lining. These cells release CCK into the bloodstream when they detect specific nutrients, especially free fatty acids and certain amino acids. Physiologically, CCK is a real multitasker. It's crucial for regulating gastric emptying, stimulating gallbladder contraction, to release bile, relaxing the sphincter of Oddi, which lets bile and pancreatic juice into the duodenum, and boosting pancreatic enzyme secretion. And recent research suggests CCK acts both directly on the stomach to inhibit emptying and indirectly by stimulating those vagal nerve fibers, initiating a reflex that also helps slow things down.
Okay, that perfectly explains how things slow down. But if all these inhibitory signals are going, how does emptying ever continue? How does the stomach keep sending more chyme down?
Ah, that's the elegance of this dynamic balance. As the chyme moves further down the small intestine, specifically into the next part, the jejunum, the strength of that initial feedback inhibition starts to fade. Why? Well, because there's less direct activation of those duodenal sensory mechanisms by the nutrients way down there. At this point, the pressure in the upper stomach, the intragastric pressure, it can rise again, moving more material toward that antral pump. Those powerful antral peristaltic contractions deepen once more, leading to the pylorus opening and the release of another, carefully measured amount of gastric contents into the duodenum. It's a continuous, regulated pulse.
Right. When we connect this to the bigger picture, this kind of precise control system must have immense implications for our overall health, especially thinking about conditions like obesity.
Absolutely. The anthoclinic section from our source highlights this perfectly. The entire GI tract plays a major role in sensing nutrients and signaling satiety, that feeling of fullness. It activates neural and endocrine pathways that communicate with other systems, like our fat energy stores, to meticulously regulate energy balance. Hormones like CCK, which we mentioned, but also GLP-1, glucagon like peptide 1, and PYY, peptide YY, are well-known satiety hormones. They tell the brain you've had enough. We see this in clinical practice too. Giving PYY externally can inhibit food intake in both lean and obese individuals. And a GLP-1 analog, exenatide, is actually used as a weight loss medication. And bariatric surgeries, things like Roux-en-Y gastric bypass or vertical sleeve gastrectomy, they achieve pretty substantial and lasting weight loss. This is partly due to the smaller stomach pouch leading to earlier satiety, sure, but also significantly by beneficially altering these gut hormone profiles. Even effects on bile acids and the gut microbiome are now recognized as contributing factors to their success. It's fascinatingly complex.
Okay, so with the chyme now precisely delivered into the small intestine, it's time for some serious chemical breakdown, and this is where the exocrine pancreas steps in, providing what's truly the largest contribution of digestive enzymes, right?
It's a truly impressive organ, yes.
Yeah.
Beyond its crucial enzymes, the pancreas provides vital substances like water and, very importantly, bicarbonate ions. These are absolutely essential for neutralizing the highly acidic gastric chyme coming in. They ensure the small intestinal lumen maintains a near-neutral pH, somewhere close to 7.0. And this is critical for two main reasons. One, pancreatic enzymes are just inactivated by high acidity. And two, a neutral pH also protects the delicate intestinal lining from acid and the enzyme pepsin.
So the pancreas is kind of like the ultimate pH balancer for the gut.
Precisely. While, yes, biliary ductules and duodenal cells chip in, the pancreas is by far the largest provider of bicarbonate. Structurally, the pancreas is organized a bit like salivary glands, with these clusters of secretory cells called acini, which drain into a branching system of ducts. The acinar cells produce the initial primary secretion, a fluid rich in organic stuff, particularly digestive enzymes, with an ionic composition much like blood plasma. And this acinar fluid secretion is primarily stimulated by CCK.
And then the ducts sort of refine this pancreatic juice.
Yes, exactly. As this primary secretion flows through the pancreatic ducts, it gets diluted and made more alkaline. Chloride ions are reabsorbed and bicarbonate is added. The adult human pancreas produces about, 1.5 liters of this pancreatic juice daily? That's quite a bit. Our source details its major constituents. You've got inactive precursors of proteases like trypsinogen, chymotrypsinogen, starch-digesting amylase, lipid-digesting enzymes like lipase, nucleases, and important regulatory factors too, like procolipase and these crucial trypsin inhibitors.
Inactive precursors for proteases? That sounds like an incredibly smart safety mechanism. The pancreas doesn't want to digest itself, I imagine.
Oh, it's absolutely critical for self-preservation. Many digestive enzymes, especially the proteolytic ones, the protein breakers, are produced as these inactive proforms.
Yeah.
This design prevents the pancreas from, digesting its own tissues. And there are multiple layers of protection. Pancreatic juice contains various trypsin inhibitors to reduce this risk because trypsin is the master activator for all the other proenzymes. There's even a second layer of defense. Trypsin molecules can actually degrade other trypsin molecules. That's quite clever.
And what happens when that sophisticated safety system does fail? You're hinting at pancreatitis, aren't you? At the cellular level box explains this consequence.
That's exactly right. Pancreatitis occurs when these powerful enzymes activate prematurely within the pancreas itself before they reach the relative safety of the small intestine. In hereditary pancreatitis, for instance, patients might have mutations in the trypsin enzyme, making it resistant to degradation, or mutations in the trypsin inhibitors, making them inactive. If these defenses are breached and trypsin becomes active too soon, you get this vicious cycle of enzyme activation, leading to those painful, often dangerous bouts of pancreatitis.
Wow. That's a stark reminder of how essential these precise controls are for keeping us healthy. Let's delve deeper into those controls, especially that critical pH regulation system.
Yeah, this self-limited pH regulatory system is truly elegant. When the pH in the small intestinal lumen drops below about 4.5, specialized endocrine cells in the intestinal lining, called S cells, they get triggered. They respond to this increased acidity, the H+ ions, by releasing the hormone secretin, and it's a classic negative feedback loop. As secretin prompts bicarbonate secretion, the luminal pH rises, which then, in turn, shuts off the secretin release signal. Beautifully simple, really.
So secretin is the signal. What's its direct effect once it's released into the bloodstream?
Okay, so secretin primarily stimulates the epithelial cells lining the pancreatic ductules. It tells them to secrete bicarbonate into the lumen, the duct's channel, and water follows passively to maintain osmotic equilibrium. At the cellular level, what happens is secretin increases cyclic AMP, or CAMP, inside these ductular cells. This CAMP increase opens specific chloride channels called CFTR channels. That stands for cystic fibrosis transmembrane conductance regulator. This causes an outflow of chloride ions into the duct lumen. This chloride outflow then drives an adjacent antiporter protein, which exchanges those chloride ions for bicarbonate ions, bringing bicarbonate into the lumen. The CFTR channel itself is also directly permeable to bicarbonate. So it's a couple of mechanisms working together, complex ion transport pathways, but absolutely essential for buffering that incoming acid.
And that bicarbonate has to come from somewhere, right? It doesn't just appear.
Precisely. The bicarbonate used in this process comes from two main sources. Some of it is actively taken up across the basolateral membrane, the backside of the ductular cells, by a transporter called NBC1, the sodium bicarbonate cotransporter type 1. And interestingly, a portion of this bicarbonate actually originates from the circulating bicarbonate produced during gastric acid secretion elsewhere in the body, a neat example of systemic integration. The rest can be generated right there, inside the ductal cells, by the enzyme carbonic anhydrase. The net effect is a significant increase in both the pH and the volume of pancreatic juice delivered to the intestine.
This immediately makes me think of cystic fibrosis, of course. The "in the clinic" section from our source ties right into this, showing how devastating a CFTR mutation can be for this whole system.
Indeed. Cystic fibrosis, the genetic disease, it impacts CFTR function in numerous epithelial organs: lungs, intestine, biliary system, and, critically here, the pancreas. The mutation impairs the ability of these organs to properly hydrate and alkalinize their luminal contents. In the GI system specifically, this can lead to severe issues like intestinal obstruction, damage to the duodenum, liver, and biliary problems, plus that pancreatic dysfunction. Many CF patients need lifelong digestive enzyme supplements just to maintain adequate nutrition. The development of drugs specifically targeting the mutant CFTR protein has been truly revolutionary, significantly extending life expectancy for these patients. It's a major medical advance.
Okay, so secretin primarily handles the bicarbonate and fluid from the ducts. But what about the enzymes from the acinar cells? You mentioned CCK playing a key role there earlier.
Correct. While secretin is the dominant physiological activator for the ductules, CCK plays the predominant role in stimulating the acinar cells to release their payload of digestive enzymes. And understanding how CCK release itself is controlled is crucial here. CCK is produced by those I cells, classic enteroendocrine cells, embedded in the small intestinal epithelium. These cells release CCK into the surrounding interstitial space when they detect specific food components, particularly free fatty acids and certain amino acids right there in the lumen. It can be a direct interaction.
But there are also these releasing factors that kind of fine-tune this process, aren't there?
Yes, exactly. Two key luminally acting releasing factors stimulate the I cells. The first is called CCK-releasing peptide. It's secreted by other paracrine cells in the intestinal lining in response to fat and protein digestion products. The second is monitor peptide, which is actually released by the pancreatic acinar cells themselves into the pancreatic juice. Both of these, CCK-releasing peptide and monitor peptide, can also be released by neural input. That's especially important for initiating pancreatic secretion even during the cephalic phase, just thinking about or smelling food in the gastric phase when the stomach is filling. It essentially pre-prepares the system.
So these releasing factors are like highly precise volume controls for CCK, making sure the right amount of enzymes are available for the meal.
That's their primary role, precisely. to match CCK release
Yeah.
and therefore the availability of pancreatic enzymes to the meal's exact digestive needs. And because these releasing factors are peptides themselves, once the meal has been fully digested and absorbed, they get degraded by proteases. This elegantly shuts off the CCK release signal, preventing unnecessary enzyme production. It's a really neat self-regulating feedback loop designed for efficiency.
And once CCK is released, how does it then trigger the acinar cells to actually release their stored enzymes?
All right, CCK works through a dual mechanism here. First, as a classic hormone, it travels through the bloodstream and binds to specific CCK receptors located directly on the pancreatic acinar cells. Second, it also stimulates crucial neural reflex pathways that influence the pancreas. Vagal afferent nerve endings in the wall of the small intestine also have these CCKA receptors. When CCK binds to these nerve endings, it activates a vagovagal reflex. This reflex further enhances acinar cell secretion by activating pancreatic and enteric neurons and stimulating the release of key neurotransmitters like acetylcholine, GRP, gastrin-releasing peptide, and VIP, vasoactive intestinal polypeptide.
So it's a wonderfully coordinated system using both hormonal signals and neural signals.
Indeed, a testament to coordinated biological control. The powerful digestive enzymes secreted by these acinar cells are largely synthesized and then stored in specialized granules, clustered near the apical pole atop of the acinar cells, ready for deployment.
The most potent stimuli for secretion, CCK, acetylcholine, GRP, they all act by mobilizing intracellular calcium. This calcium surge triggers a cascade, leading to the phosphorylation of various regulatory and structural proteins inside the cell. This in turn moves the granules towards the apical membrane, where they fuse and discharge their contents into the acinar lumen. These contents are then washed out by an exudative plasma and then by those ductular secretions we talked about. In the period between meals, these granule constituents are resynthesized by the acinar cells and then stored again, poised for the next meal.
Okay, we've covered gastric emptying and the vital role of pancreatic juice. Another crucial digestive secretion that mixes with the meal in the small intestine is bile. What's its primary physiological role in all this?
Right, bile. If we connect this to the bigger picture, bile is absolutely essential for aiding in the digestion and, perhaps even more critically, the absorption of lipids, or fats. It's produced up in the liver, then stored and concentrated down in the gallbladder until a meal triggers its release. And similar to pancreatic secretion, gallbladder contraction and the relaxation of that gatekeeper muscle, the sphincter of Oddi, are predominantly stimulated by CCK.
And the key components of bile that are really central to digestion are the bile acids, correct?
Yes, the bile acids are definitely the workhorses here. They're remarkable molecules because they are amphipathic. That means they have both a hydrophobic water-fearing side and a hydrophilic water-loving side. This unique structure lets them act as biological detergents. They form these tiny structures called micelles, and these micelles serve a crucial purpose. They shield the hydrophobic products of lipid digestion fatty acids, monoglycerides from the watery environment of the intestinal lumen. They essentially make fats soluble in water, which is vital for their absorption. And to get optimal lipid absorption, we need large quantities of bile acids, somewhere around 1 to 2 grams per day.
Wow, 1 to 2 grams. That's a lot of bile acids. Does the body really produce that much from scratch every single day?
This is where the body's remarkable efficiency truly shines. The vast majority of that bile acid pool is constantly recycled. It goes from the intestine back to the liver after each meal via a process called the enterohepatic circulation. Our source illustrates this cycle quite clearly. Bile acids are synthesized in a conjugated form, which is key because this limits their ability to just passively cross the intestinal lining. It keeps them right where they're needed in the lumen to help with lipid assimilation. However, once the meal reaches the terminal ileum, the very end of the small intestine, where lipid absorption is mostly done, these conjugated bile acids are actively reabsorbed by a specific transporter called ASBT, the apical sodium-dependent bile acid transporter. Then they're returned to the liver to be used all over again.
So they're not just simple detergents. They seem to have broader biological roles beyond just helping digest fat.
That's an excellent point. While their detergent role is critical, bile acids also bind to both cell surface receptors and nuclear receptors in various cell types throughout the body. This allows them to regulate not only their own synthesis, but also to influence other important metabolic processes. It really highlights their complex and systemic influence. They're signaling molecules, too.
Okay, so with the food broken down by enzymes and solubilized by bile, let's dive into the core function, taking up the products of digestion. We're talking the big 3 macronutrients, carbohydrates, proteins, and lipids. What's the fundamental principle for absorbing all of them?
Well, the fundamental principle is that macromolecules, the large molecules in our food like starches, proteins, triglycerides, they're generally just too big to be absorbed directly across the epithelial cells lining the intestine. They have to be broken down first into smaller, simpler constituents through chemical and enzymatic digestion. The small intestine isn't just crucial for absorption into the bloodstream, but also for carrying out these final stages of digestion, breaking food down into molecules simple enough to be transported across its lining.
Right. Let's start with carbohydrates then. How does the body break down complex things like starches or even table sugar into units it can actually absorb?
Okay, carbohydrate digestion happens in two main phases. First, out in the lumen of the intestine, and then directly on the surface of the enterocytes, the intestinal cells. We call that brush border digestion. Dietary carbs come in several classes. You've got starch, like amylose and amylopectin, which is a major calorie source. Disaccharides, like sucrose, table sugar, which is glucose plus fructose, and lactose, milk sugar, glucose plus galactose. And dietary fiber, which are carbohydrate polymers or human enzymes just can't digest, though gut bacteria down in the colon can ferment them. But the crucial principle for absorption is this. The intestine can only absorb monosaccharides, the simplest single sugar units like glucose, fructose, and galactose.
Okay, so the breakdown of starch is the first big step here.
Right. Luminal digestion of starch begins, technically, with salivary amylase in the mouth, though this step isn't essential for healthy adults. Maybe more important in infants or if pancreatic function is low. But the most significant player is pancreatic amylase. Both salivary and pancreatic amylase hydrolyze specific internal alpha-1,4 bonds within starch molecules. But they can't break down external bonds or the specific alpha-1,4,6 branch points found in amylopectin. So this action yields shorter glucose chains, dimers like maltose, trimers like maltotriose, and larger fragments called alpha-limit dextrins.
And then that leads us to the second really crucial phase of the brush border.
Exactly. That's the brush border digestion. This is where dietary disaccharides like sucrose and lactose and those starch oligomers from the luminal digestion get hydrolyzed into their individual monosaccharide components. This happens directly on the surface of the small intestinal epithelial cells. There's a family of membrane-bound enzymes there, heavily glycosylated, meaning they have sugar chains attached that protect them from being digested themselves by luminal proteases. Key brush border hydrolases for carbs include sucrase, assimilase, glucoamylase, and lactase. Assimilase is particularly critical for handling those alpha-limit dextrins because it's the only enzyme we have that can cleave both the alpha-1,6 and the alpha-1,4 bonds.
And what about lactase? We know its expression often changes after infancy, leading to lactose intolerance. The In the Clinic box explains that common issue.
It's exactly right. Lactase often undergoes a normal developmental decline in expression after weaning. It's particularly common in certain ethnic groups, Asians, African Americans, Hispanics, and especially if lactose isn't a consistent part of the adult diet. When there's this relative scarcity of lactase digestion, rather than its uptake, becomes the rate-limiting step for absorption. So undigested lactose stays in the intestinal lumen, it draws water in osmotically, and that leads to those uncomfortable symptoms: abdominal cramping, gas, diarrhea. Many people manage this by taking bacterial lactase enzyme supplements before eating dairy.
Okay, so once these carbohydrates are finally broken down into monosaccharides, glucose, fructose, galactose, how do they actually get into the body?
Into the cells. Yeah, this is where specialized transporters come into play, and it's really fascinating how specific this system is. These water-soluble monosaccharides have to be moved across the hydrophobic plasma membrane of the enterocyte. Glucose and galactose are taken up by a transporter called SGLT1, the sodium glucose transporter 1. It's a symporter. It couples the transport of the sugar with the sodium ion, using the sodium gradient to move the sugars into the cell, even against their concentration gradient. Fructose, on the other hand, uses a different mechanism: facilitated diffusion through the GLUT5 transporter. No sodium needed there. Then, once inside the enterocyte, all three monosaccharides, glucose, galactose, and fructose, exit the cell into the bloodstream via the GLUT2 transporter located on the basolateral membrane, the bottom side, ready to be delivered to the rest of the body.
So if one of those specific transporters, like SGLT1, isn't working correctly, that could cause pretty significant problems with nutrient absorption.
That's a critical point. The In the Clinic section on glucose-galactose malabsorption highlights precisely this scenario. It's a rare genetic disorder caused by mutations in the SGLT1 gene. These mutations might result in a faulty or even completely unexpressed SGLT1 protein, or more commonly, a failure of the protein to get to the apical membrane where it needs to function. The malabsorbed glucose, unable to be taken up, stays in the gut, draws water in, and contributes to severe diarrheal symptoms, especially in infants. Despite being rare, studying this condition provided absolutely crucial insights into how intestinal epithelial transport fundamentally works.
Okay, proteins are next, and given their complex structures with 20 different amino acids, this seems like maybe a more intricate challenge to the body.
Indeed, proteins are quite complex, and while the body, particularly the liver, has a significant ability to interconvert many amino acids, there are certain essential amino acids that we simply cannot synthesize internally. They must be obtained directly from our diet. Our source highlights these essential ones.
So where does the breakdown of proteins actually begin?
Right, protein assimilation involves three distinct phases of enzyme-mediated digestion. The first phase happens up in the gastric lumen, inside the stomach. Here, the enzyme pepsin gets to work. Pepsin is released from chief cells as an inactive precursor called pepsinogen. In the highly acidic environment of the stomach, pepsinogen autocatalytically cleaves itself to yield the active enzyme, pepsin. Pepsin is specialized for this acidic environment, and it cleaves proteins at specific sites, usually involving neutral amino acids. But it's important to note, pepsin isn't capable of fully digesting protein into absorbable forms. What it yields is a mixture of some intact protein, a large majority of sizable peptides, and only a limited number of free amino acids.
Okay, so it starts the job, but then it's on to the small intestine for the main event of protein digestion.
Exactly. The partially digested protein coming from the stomach then encounters the powerful pancreatic proteases down in the small intestine. Crucially, these are also secreted in inactive forms, remember, to prevent damage to the pancreas itself. Their activation is carefully delayed until they reach the small intestinal lumen. This activation cascade is initiated by enterokinase, an activating enzyme found only on the brush border of the small intestinal epithelial cells. Enterokinase cleaves trypsinogen to yield active trypsin, and trypsin then acts as the master activator, cleaving all the other protease precursors secreted by the pancreas chymotrypsinogen, proelastase, procarboxypeptidase A, procarboxypeptidase B. This results in a powerful, diverse mixture of active enzymes that can pretty much completely digest the vast majority of dietary proteins.
And what kinds of enzymes are these, functionally speaking? How do they attack the proteins?
Well, trypsin, chymotrypsin, and elastase are examples of endopeptidases. This means they cleave internal bonds within the peptide chain, breaking long chains into shorter peptides, rather than snipping off single amino acids from the ends. For instance, trypsin specifically cleaves proteins next to basic amino acids like lysine and arginine, resulting in peptides that have a basic amino acid at their C-terminus. These shorter peptides are then acted upon by pancreatic ectopeptidases like carboxypeptidase A and B. These specialize in cleaving single amino acids from the very end, the C-terminus, of a peptide chain.
Okay, and the final stage of protein digestion happens right there at the intestinal lining itself.
Precisely. That takes place at the brush border of the enterocytes. Mature intestinal cells express a whole variety of membrane-bound peptidases. These include both aminopeptidases, which snip from the N-terminus, and carboxypeptidases, snipping from the C-terminus. These further break down peptides into products small enough for uptake across the apical membrane. But even with this substantial enzymatic arsenal, some dietary peptides, especially those containing tricky amino acids like proline or glycine, can be resistant to hydrolysis and are digested very slowly. Fortunately, the intestine has this remarkable capability to take up short peptides, specifically dipeptides and tripeptides, in addition to single amino acids. The majority of these intact peptides taken into the enterocyte are then subjected to a final stage of digestion inside the cell's cytosol, liberating their constituent amino acids. These can then be used by the cell itself or exported for the rest of the body. A small proportion of Diababe's tripeptides might even be transported intact directly into the bloodstream.
So how do these end products, the amino acids and those short peptides, then get absorbed into the body?
Okay, so the body is equipped with a sophisticated series of plasma membrane transporters designed to facilitate the uptake of these water-soluble products. Given the large number of different amino acids, there's a relatively extensive number of specific transporters, usually handling subsets of amino acids like neutral ones, anionic ones, cationic ones, though there can be some overlap in what they carry. Furthermore, some, but not all of these amino acid transporters are symporters, coupling transport with sodium ions using that gradient. The small intestine is also really notable for its ability to take up those short peptides. The primary transporter responsible for this is called PepT1, peptide transporter 1. It's a supporter that transports peptides along with protons, H+ ions. Once inside the cell, any amino acids liberated from these peptides that aren't immediately needed by the enterocyte itself are then exported across the basolateral membrane into the blood capillaries, heading straight to the liver via the portal vein.
This whole system, especially for amino acid uptake, sounds incredibly robust, almost like it has built-in redundancies, backups.
That's an excellent observation, and a key insight highlighted at the cellular level section of our source. This redundancy and uptake mechanisms for protein digestion products really underscores how critical protein assimilation is. It also means that even if there are deficiencies in specific amino acid uptake pathways across the intestine, it often doesn't result in actual nutritional deficiencies. For example, in cystinuria, where the amino acid cysteine is poorly reabsorbed from the urine, leading to kidney stones, nutritional deficiencies typically don't occur from the gut because other uptake mechanisms can compensate. Similarly, in Hartnuff's disease, caused by mutations in a specific neutral amino acid transporter, SLC6A19, there's a loss of neutral amino acids in the urine, leading to psychiatric and neurological issues, but usually not nutritional deficiencies per se, again because of compensatory pathways like PEPT1. And PEPT1 itself is clinically quite interesting because it can mediate the uptake of certain peptidomimetic drugs, drugs designed to look like peptides, including some antibiotics and even cancer chemotherapeutic agents. It's a fascinating application of understanding this pathway.
Okay, our third macronutrient class, lipids or fats. These are defined by being more soluble in organic solvents than water, and of course they pack a significant caloric punch.
They are indeed of major nutritional significance. They supply more calories per gram than either proteins or carbohydrates. The predominant form in our diet is triglyceride, composed of long-chain fatty acids. That means carbon chains longer than 12 carbons esterified to a glycerol backbone. We also consume phospholipids and cholesterol, mostly from cell membranes and also from bile itself. And crucially, the fat-soluble vitamins A, D, E, and K are essential nutrients. They're present in tiny amounts, but they're almost totally insoluble in water, which means they require very special handling by the body to ensure their uptake.
So the first big challenge with fats is their water insolubility. How does the body even prepare them for digestion and absorption in the watery gut?
That's the critical first question. When a fatty meal is ingested, the lipid becomes liquefied at body temperature, right? And it naturally floats on top of the watery gastric contents. This would severely limit the surface area, the interface between the watery environment and the lipid phase, restricting enzyme access. So an early and absolutely critical stage in lipid assimilation is emulsification. The stomach's powerful mixing action churns the dietary lipid into a suspension of incredibly fine droplets, vastly increasing the surface area where enzymes can actually work. This process is then greatly facilitated later by the formation of mixed micelles with the aid of those bile acids supplied in biliary secretions.
Okay, so the fats are emulsified into tiny droplets and then solubilized by bile acids. Now, how does the actual chemical digestion process happen?
Right. Lipid digestion technically begins up in the stomach with gastric lipase, an enzyme released from gastric chief cells. It absorbs to the surface of these tiny fat droplets and starts hydrolyzing triglycerides into diglycerides and free fatty acids. However, it's worth noting that in healthy individuals, gastric lipolysis is largely dispensable. It's not essential because of the marked excess of pancreatic enzymes that will soon take over the job. The majority of significant fat digestion actually occurs down in the small intestine.
Okay, so which pancreatic enzymes are the key players here for lipids?
Pancreatic juice contains 3 crucial lipolytic enzymes, all optimized for activity at that neutral pH we established earlier. First, and most important, is pancreatic lipase. This enzyme differs from gastric lipase because it can hydrolyze both the 1 and 2 positions of a triglyceride molecule, yielding free fatty acids and monoglycerides. Now, here's a key insight. Pancreatic lipase itself actually appears to be inhibited by bile acids, which also adsorb to the surface of the oil droplets and can cause lipase to detach. Seems counterintuitive, right? But its activity is miraculously sustained by an essential cofactor called collapase, also supplied in pancreatic juice. Collapase acts like a bridging molecule. It binds to both the bile acids and the lipase enzyme, effectively anchoring lipase to the oil droplet even in the presence of those inhibitory bile acids. It's an incredibly smart design.
That is ingenious. What are the other 2 enzymes involved?
Okay, pancreatic juice contains 2 more important enzymes for fat digestion. One is phospholipase A2, which hydrolyzes phospholipids like those found in cell membranes. Predictably, this enzyme would be quite toxic if active inappropriately, so it's secreted as an inactive proform, only activating once it reaches the safety of the small intestine. The third is a relatively non-specific cholesterol esterase. It can break down esters of cholesterol, fat-soluble vitamins, and even triglycerides. Interestingly, this enzyme requires bile acids for its activity, unlike lipase. It's also closely related to an important enzyme found in breast milk that plays a significant role in fat digestion in newborns.
So as this lipolysis, this breakdown proceeds, the products, the fatty acids and monoglycerides, need to somehow get from the middle of that fat droplet to the absorptive surface of the intestinal cell. How does that happen?
Correct. The products of digestion are abstracted, pulled away from the lipid droplet. First, they go into a sort of lamellar or membrane-like phase, and then subsequently into those mixed micelles we talked about earlier. These micelles are composed of the lipolytic products themselves, as well as bile acids. The amphipathic bile acids expertly shield the hydrophobic regions of the lipid products from the water, while presenting their own hydrophilic faces to the aqueous environment. These micelles are truly in solution, which means they dramatically increase the solubility of these lipid products in the watery intestinal contents. This significantly boosts the rate at which lipids can diffuse across the unstirred water layer to reach the absorptive epithelial surface.
So are micelles absolutely essential for all fat absorption or just certain types?
That's a critical question. While they are indeed crucial for cholesterol and fat-soluble vitamins, which are almost totally water-insoluble, a key insight here is that micelles are not absolutely essential for triglyceride absorption. Why? Well, thanks to the immense surface area of the small intestine and the appreciable, albeit low, molecular solubility of the triglyceride hydrolysis products themselves, fatty acids and monoglycerides. So patients with insufficient bile acid output, maybe due to a gallstone obstructing bile flow, don't typically experience severe fat malabsorption, meaning problems absorbing triglycerides. However, because cholesterol and fat-soluble vitamins are almost totally insoluble in water, they do absolutely require micelles for absorption, even after digestion. If luminal bile acid concentrations fall below what we call the critical micellar concentration, these patients can easily become deficient in fat-soluble vitamins.
Okay. Given that lipids are naturally lipophilic, fat-loving, do they just passively diffuse across the cell membranes, or is there more regulation to their uptake?
Well, while their lipophilicity certainly allows them to cross membranes quite readily compared to water-soluble molecules, recent evidence strongly suggests that their uptake isn't just simple diffusion. It appears to be regulated, or at least facilitated, by specific membrane transporters. For instance, a protein called MVM-FABP, microvillus membrane fatty acid binding protein, seems to facilitate the uptake of long-chain fatty acids across the brush border. Similarly, a protein called NPC1L1 Niemann-Pick-C1-like 1 has been identified as a significant uptake pathway for cholesterol into enterocytes. And it's now a therapeutic target for people with hypercholesterolemia, those high cholesterol levels. However, cholesterol uptake overall is surprisingly inefficient. Why? Because cholesterol, along with plant sterols from our diet, can also be actively pumped back out of the enterocytes into the lumen by a complex of two ABC transporters, specifically ABCG5 and G8. It's like a revolving door. Finally, the glycerol backbone released from triglycerides can be transported into intestinal epithelial cells by specific channels called aquaglyceroporins.
So drugs can specifically target these transporters, like NPC1L1, to influence lipid absorption.
That's absolutely correct, and the in the clinic section confirms this clinical relevance. Ezetimibe, a drug specifically targeting NPC1L1, reduces cholesterol absorption. It prevents dietary cholesterol uptake and can also disrupt its enterohepatic circulation. Clinical studies show that ezetimibe synergistically improve the effectiveness of other strategies used to lower circulating levels of LDL cholesterol, the bad cholesterol, such as statin drugs. This is particularly important for those at risk for cardiovascular disease. It really illustrates the precision with which we can now intervene based on this molecular understanding.
So once the lipids are absorbed into the enterocyte, what happens to them then? This pathway seems quite different from how carbohydrates and proteins are handled after absorption.
Yes, this is a crucial distinction and a key difference in lipid metabolism. Unlike monosaccharides and amino acids, which leave the enterocyte pretty much as they entered and go straight into the portal blood circulation, the products of lipolysis take a different route. They are re-esterified within the enterocyte, specifically in the smooth endoplasmic reticulum, to reform triglycerides, phospholipids, and cholesterol esters. Basically, they're rebuilt. Concurrently, the enterocyte synthesizes a series of specialized proteins known as apolipoproteins in the rough ER. These proteins are then combined with the resynthesized lipids to form a complex structure called a chylomicron. Think of it as a tiny package, a lipid core coated by these apolipoproteins.
Okay, chylomicrons. And where do these packages go once they're assembled inside the cell?
Right, these chylomicrons are then exported from the enterocyte via a process called exocytosis. They butt off the cell. But here's the critical difference. Once they enter the lamina propria, the connective tissue layer beneath the intestinal lining, they're simply too large, ranging from 750 to 5,000 angstroms in diameter, to squeeze through the gaps between the cells of the mucosal capillaries. Instead, they are taken up into the lymphatic vessels, the lacteals, within the lamina propria. This means they bypass the portal circulation entirely, and at least for their first pass through the body, they bypass the liver. Eventually, these chylomicrons floating in the lymph fluid enter the general bloodstream via the thoracic duct near the heart. They serve as the primary vehicle to transport dietary lipids around the body for use or storage by cells and other organs.
Are there any exceptions to that whole chylomicron pathway for lipid transport?
Yes, medium-chain fatty acids, those with shorter carbon chains, are a notable exception. They are relatively more water-soluble than long-chain fatty acids, and unlike their longer counterparts, they can actually permeate the tight junctions between enterocytes quite appreciably. This means they largely bypass that complex intracellular processing and chylomicron packaging we just described. They therefore enter the portal circulation directly, just like sugars and amino acids, and are more readily available to tissues like the liver. For patients who have trouble with bile acid production or micelle formation, a diet rich in medium-chain triglycerides can be particularly beneficial for energy intake, as their absorption is less dependent on that whole process.
Okay, digestion and absorption clearly require a constantly moving, watery environment. Let's talk about the incredible volumes of fluid that move through our gut daily. It's truly astonishing when you look at the numbers.
It really is. The fluidity of intestinal contents, particularly in the small intestine, is absolutely vital. It's needed for the efficient propulsion of the meal along the tract and to allow digested nutrients to diffuse effectively to their absorption sites at the cell surface. Consider this: while we ingest only about 1 to 2 liters of fluid daily from food and drink, an astounding additional 8 liters are supplied by secretions from the stomach, small intestine, pancreas, and liver combined. So the small intestine is presented with a staggering 9 to 10 liters of fluid every single day.
That is a huge amount, 10 liters. But thankfully, we don't excrete anything close to that, so the vast majority must be absorbed back.
Precisely. In healthy individuals, only about 2 liters of this massive fluid load actually pass from the small intestine into the colon for further reabsorption, and eventually only a mere 100 to 200 milliliters exits the body in stool. So yes, the intestine overall is an incredibly powerful water-absorbing organ. During a period after a meal, this absorption in the small intestine is primarily driven by the osmotic effects of nutrient absorption itself. As nutrients like glucose and amino acids are actively transported into the cells, they create an osmotic gradient across the intestinal lining. This gradient then passively drives the movement of water across the tight junctions between the cells, following the solutes.
So the small intestine is a big net absorber of water, but it also actively secretes fluid too, doesn't it? It's not just one-way traffic.
That's a critical observation, yes. Even though the net water and electrolyte transport in the small intestine is predominantly absorptive overall, the tissue definitely engages in active electrolyte secretion as well. This secretion is carefully regulated. It happens in response to various signals, including the composition of the luminal contents, mechanical deformation of the mucosa, like stretching, and intestinal distension. Key secretagogue substances that stimulate secretion include neurotransmitters like acetylcholine and VIP, as well as signaling molecules like prostaglandins and serotonin. This secretion is essential. It ensures that intestinal contents remain appropriately fluid while digestion and absorption are ongoing. It's also important for lubricating the passage of food particles along the length of the intestine.
And when this secretion is abnormally low, that can lead to significant clinical problems, like constipation.
Exactly. Clinical evidence strongly suggests that conditions like constipation and even intestinal obstruction, the latter being common in cystic fibrosis, can result, at least in part, from abnormally low intestinal secretion. The majority of the intestinal secretory flow of fluid into the lumen is driven by the active secretion of chloride ions, often via that CFTR channel we discussed. In cystic fibrosis, the loss of functional CFTR chloride channels leads to these issues of thickened mucus and obstruction, although accessory chloride channels like TM16A can sometimes partially compensate.
And bicarbonate secretion also occurs in certain specialized areas.
Yes, that's another important specialized function. Some segments of the intestine, particularly the most proximal portions of the duodenum, right after the stomach, engage in active bicarbonate secretion. This is a crucial protective mechanism. It shields the delicate duodenal epithelium from damage caused by the highly acidic chyme and the digestive enzyme pepsin entering directly from the stomach.
What about other essential nutrients like vitamins and minerals? Are they also handled by specific mechanisms in the small intestine?
Absolutely. The small intestine is also the crucial site for absorbing water-soluble vitamins, such as vitamin C and cobalamin, vitamin B12, and essential minerals like calcium, magnesium, and iron. In general, these substances are taken up from the luminal contents via the activity of specific transporters, each tailored for particular nutrients. Vitamin B12 absorption involving intrinsic factor is a classic example. And importantly, their uptake is also precisely controlled, often by sophisticated feedback mechanisms. These systems sense the concentration of the relevant substance already circulating in the bloodstream and adjust the expression of these intestinal transporters and accessory molecules accordingly. It's another layer of the body's meticulous management of its internal environment.
Okay, finally, let's talk about the mechanics of the small intestine. The smooth muscle layers create very specific motility patterns to mix and move everything along. What are they, and how do they differ dramatically between the fed state after eating versus the fasted state?
Right. That's a critical aspect of intestinal function. And it's truly remarkable how these patterns perfectly adapt to the body's current needs. In the postprandial or fed period, the primary goal of motility is really to mix and retain the meal within the small intestine. This allows for thorough digestion and maximal contact time for absorption. This consists largely of what we call segmenting contractions and also some retropulsive movements, both designed to kind of slow down transit and churn the contents.
So segmentation is primarily about mixing the contents, not necessarily pushing it forward.
Yes, precisely. Segmentation is a stereotypical pattern. It involves rhythmic, localized contractions of the circular muscle layer that effectively divide the intestine into small segments. This reflects programmed activity of the enteric nervous system, the gut's own brain. It effectively churns the intestinal contents back and forth, ensuring optimal mixing with digestive secretions and maximal exposure of the nutrients to the absorptive surface of the intestinal lining. While peristalsis, those wave-like contractions that do propel contents distally, also occurs in the fed state, it's generally less dominant than segmentation. Peristalsis involves that coordinated contraction behind a bolus of food and relaxation ahead of it, designed to move material forward.
And then, when you haven't eaten for a while, the gut switches gears entirely to a different kind of housekeeping system.
Absolutely. During fasting periods, the motor patterns become organized into a very distinct cyclical sequence of phases known as the migrating motor complex, or MMC. You can think of this as essentially the gut's housekeeper, or the intestinal broom. It's designed to sweep any remaining gastric and intestinal contents, undigested food particles, shed cells, mucus, bacteria out of the small intestine and down into the colon, preparing for the next meal.
How exactly does this migrating motor complex work? What are its phases?
It has pretty distinct phases. Phase 1 of the MMC is characterized by a period of relative quiescence, very little motor activity. Then, small, somewhat disorganized contractions begin during Phase 2. But the most striking part is Phase 3, which lasts for about 10 minutes or so. During Phase 3, you get these large, intense, propagating contractions that sweep along the entire length of the small intestine. These powerful contractions are stimulated by the hormone Motility, and they effectively sweep all undigested material down into the colon. Crucially, during this Phase 3 activity, both the pylorus, at the stomach exit, and the ileocecal valve, the valve between the small and large intestines, fully open. This allows even large, undigestible items that might have been ingested to pass out of the small intestine.
And what happens to this migrating motor complex, this housekeeping wave, when you actually ingest a meal?
The ingestion of a meal immediately suspends the MMC. It stops it in its tracks. Motility levels in the blood fall dramatically, and the complex cannot resume its cycle until these levels rise again, usually after the meal has been substantially processed. The entire cycle of the MMC typically takes about 90 minutes in healthy adults during fasting.
So after all that, what does this all mean for us, the listeners? We've just taken an incredibly deep dive into the small intestine, from how it meticulously manages food flow from the stomach, to the amazing array of enzymes and chemicals it gets from the pancreas and liver, to the intricate ways it breaks down and absorbs every last carbohydrate, protein, and fat. And we've seen how it masterfully manages these huge fluid volumes and moves contents along with such precise motor patterns.
Yeah, if we connect all this back to the bigger picture, it truly underscores the extraordinary biological engineering at play here. Every single aspect, from the microscopic villi maximizing surface area to those sophisticated feedback loops regulating enzyme release and motility, is just exquisitely designed for maximum efficiency in nutrient assimilation and maintaining overall metabolic homeostasis. Understanding these fundamental mechanisms provides absolutely crucial insight into our overall metabolic health and precisely how the body maintains its energy balance day in, day out. You've really gained a professional-level understanding of this vital organ today.
It really has highlighted just how profoundly integrated our bodily systems are. And for you, the listener, maybe think about this. Knowing now the immense sophistication and precision of these processes happening constantly within your own small intestine, what surprising new connections might you start to make? Connections between its function and other aspects of your health. Or perhaps consider how things like specific diets, changes in your gut microbiome, or even certain medications might subtly or perhaps dramatically impact this remarkable internal world we all carry within us. It truly is a testament to biological complexity.